The disease topic. A pathogen is a microorganism which causes disease. Two types of disease include communicable diseases and non-communicable diseases. Now, communicable diseases may be transmitted from one person to another, and they are caused by pathogens. Pathogens include microorganisms like bacteria, viruses, and fungi. Non-communicable diseases cannot be transmitted from one person to another, and they are not caused by pathogens. There are various stages involved in the course of a disease. First step is infection. This is when the pathogen enters the host. The second stage is incubation, when the pathogen reproduces inside the host organism. And then the third stage is symptoms, which is when the pathogen damages cells and produces toxins, which is responsible for making the individual feel unwell. Why don't you tend to feel the symptoms of a pathogen as soon as they enter your body? Well, first up, only few pathogens enter in the first place. It takes time for them to reproduce, known as the incubation time, and it's only when you have large numbers of pathogens that you can really feel the symptoms of the disease they cause. I've already said that a virus is an example of a pathogen. Remember, viruses are non-living, they don't obey Mrs. H. Gren, they don't respire, they don't carry out homeostasis, they're not sensitive, etc. But if you think about the structure of a virus, it consists of a protein coat which surrounds either RNA or DNA. The protein coat sometimes has protein spikes on the outside of it, and enzymes are present such as reverse transcriptase, which is present in the HIV virus that causes DNA to be made from RNA. Another category of virus is the RNA virus. This consists of RNA, it contains no DNA whatsoever. Remember that viruses are known as parasites. This is because they infect a host and always cause harm to them. It's much more difficult to study viruses compared with bacteria, simply because viruses are much smaller and have far less complex machinery compared with a bacterial cell. The fact that a virus always has to reproduce inside a host also makes it difficult to study them. So we've said that viruses don't obey Mrs. H. Gren. Remember to watch the start of my all-in-one video if you're not sure what I mean by that. So how do viruses replicate? Well, first of all, viruses must infect the host organism. The virus particles attach to a host cell and inject either their RNA or DNA into the host cell. That will depend on the type of virus they are. The virus RNA or DNA is incorporated into the host's own DNA, and as the host replicates its own DNA, it replicates the viral RNA or DNA. Protein synthesis occurs. That is required in order to make more protein coats for the viruses. Lots of new viruses are produced. There's lysis of the host cell, which means that it bursts open and viral particles are released into the rest of the body. Lots of diseases are caused by viruses. Examples include polio, measles, mumps and rubella, HIV, which stands for human immunodeficiency virus. This causes the disease AIDS, as well as the common cold. Ebola is also caused by a virus. If we turn our attention to Ebola now, let's talk about the various ways in which it may be transmitted. It's through bodily fluids, so that could be saliva, blood, semen and feces. If you come into contact with any of these bodily fluids that contain Ebola, you may get the disease. Various symptoms occur when you're infected with Ebola, including diarrhea, muscle pain, internal and external bleeding, and that's one of the horrific symptoms of Ebola is bleeding from your orifices, so that could be your eyes, your ears, your anus. It's very difficult to prevent the spread of Ebola. One way includes using disinfectant to clean surfaces, to avoid contact with an infected person. If the person dies, it's to ensure safe burial, isolated burial so people can't come in contact, and to wear protective goggles and clothing. Unfortunately, there's no effective vaccine against Ebola. Oral rehydration therapy may assist with the symptoms. The reason for this is because a person suffering from Ebola does get diarrhea and vomiting, so oral rehydration therapy can help replace any lost fluids and salts. The reservoir of a pathogen is an organism which carries the pathogen without suffering any symptoms. A good example is bats, which are a reservoir of the Ebola virus. So they carry the Ebola virus without suffering any ill effects. More detail now on HIV, which I've told you stands for human immunodeficiency virus. Later on, this can cause the disease AIDS, which stands for acquired immune deficiency syndrome. So what happens with HIV is the HIV virus infects lymphocytes. The virus itself contains reverse transcriptase. So although the virus contains RNA as opposed to DNA, the reverse transcriptase enzymes causes the host cell to make viral DNA from its RNA. The type of cell infected is known as a lymphocyte. Remember, this forms part of the body's immune response to kill pathogens, and the specific type of lymphocyte infected is known as T helper cells. When the reverse transcriptase causes viral DNA to be made from RNA, that DNA is incorporated into the T helper cell's DNA. The HIV virus then instructs the T helper cell to make HIV proteins, and then as a result, the HIV proteins and RNA are assembled into more virus particles that when the cell undergoes lysis, it bursts open and releases them into the bloodstream. So how may HIV be transmitted? Well, through bodily fluids, so unprotected sex, the transfer of semen, using dirty, unclean needles, as the blood harbours HIV virus, and if you're really unlucky, infected blood transfusions. Initially, the symptoms are very mild, they're flu-like. It takes quite a large number of years for the disease AIDS to develop. The real issue with AIDS is because the virus infects T helper cells, which are lymphocytes. Remember I said that forms part of the body's response against pathogens. So it means that if you get infected with the common cold, your body is less able to fight it. And that's what actually causes the death of AIDS patients, is the buildup of all these other diseases which can't be fought against effectively. To avoid the transmission of HIV, condoms should be worn, clean needles should be used, and blood transfusions must be screened to make sure they don't contain any pathogens. Antiviral drugs can be used to prevent transmission of HIV and to slow the development of AIDS. There is no current vaccine available. Moving on to a different type of pathogen now, the bacterial cell. Remember, these are prokaryotic because they do not contain a distinct nucleus. Instead, they contain a nucleoid which contains the cell's DNA. They also have small circles of DNA known as plasmids. Bacterial cells are much larger than viruses. They have cytoplasm, which contains water and dissolved substances. They have a cell membrane that allows the entry and exit of substances. They have a cell wall made of peptidoglycan. They have a slime capsule which helps protect the cell. Ribosomes enable protein synthesis, and a flagellum is a small tail used to help the bacterial cell move. Now, bacterial cells use binary fission to reproduce. They can't use mitosis. The reason for that is because they have no nucleus. So how does binary fission work? Well, the bacterial plasmids and nucleoids replicate. The cell membrane grows between the DNA copies. The cytoplasm divides and new cell walls form. And two daughter cells are formed, each with nucleoid apiece as well as many new plasmids. Now, in terms of how bacteria reproduce and how they grow, they'll follow a specific bacterial growth curve, which starts with the lag phase. This is when there are small numbers of bacterial cells present, due to the bacteria adjusting to their new environment, so you'll see a slow rate of growth. The growth phase, known as the exponential phase, you'll see a rapid increase in bacterial numbers as they are now used to their new environment and they have a plentiful supply of oxygen and nutrients. Then the stationary phase occurs when amount of replication equals the death rate of the bacteria. And then finally the death phase, where bacterial numbers rapidly drop off due to a buildup of toxins and the fact that they're running out of oxygen and nutrients. You see mass death of the bacteria in the death phase. You may be aware that antibiotics are only effective against bacteria. They don't work against viruses, and that's because of the way in which the antibiotics work. Now, antibiotics work on the bacteria cell wall. They cause it to burst open by osmosis. Because the viruses have a protein coat, antibiotics are ineffective against them. It's possible to discern how effective antibiotics are by using a petri dish. So in this case, you have an aseptic technique, which means it will be sterilized. There'll be no bacteria present. You then use a swab or an inoculating loop to swab the agar jelly with your bacterial sample. You place your antibiotic discs on the petri dish and then you seal your dish with tape. The antibiotics, which are most effective against the bacteria, will see the largest clearance zone around them, and you can actually measure that with a ruler, find out the diameter of the clearance zone and use pi(r²) to work out the zone of inhibition. The larger the zone of inhibition, the more effective the antibiotic, because it's killed all the bacteria present. The agar jelly provides nutrients. You need sterile conditions to prevent the growth of pathogenic bacteria, so bacteria that you don't want present in your experiment. You're going to incubate your petri dish at a suitable temperature, such as 25 degrees Celsius, which enables your bacterial swab to replicate nice and quickly. A very nasty disease now, cholera, caused by the bacterium Vibrio cholerae, How cholera is spread is through infected feces. So someone suffering from the disease poops and their feces go into water, and then if you drink any water with those feces in it, you are likely to get cholera. The problem with cholera is that it causes super intense diarrhea to the point where you produce stools called rice water stools. They're very characteristic, white and cloudy in colour. The person becomes massively dehydrated, and that's actually what causes their death. To avoid getting cholera, you obviously need to be drinking clean water which doesn't contain the bacterium. Good sanitation methods need to be used where that sewage is appropriately treated, and you need to wash your hands after using the bathroom. Treatment options include antibiotics used to kill the bacteria and oral rehydration therapy used to replace lost salts and water from the chronic diarrhea. Gonorrhea is an STI, a sexually transmitted infection. It's caused by a bacteria and it's spread through unprotected sex, so a good way to avoid getting gonorrhea is by using condoms, which provide a barrier. Symptoms include discharge from the penis whilst urinating, and if it gets so bad, it can actually lead to infertility. Anyone suffering from gonorrhea is often given antibiotics, seeing as that it's a bacterial disease. Athlete's foot is caused by a fungus. This is transmitted on wet changing rooms, so you'll find that people who swim a lot might suffer from athlete's foot more than most. You end up with sore red patches usually found on your feet. That's due to your feet coming into contact with those wet changing room floors. To avoid athlete's foot, changing rooms should be dry. You should avoid walking with your bare feet on them. Keep your toenails short and use antifungal powders and creams in order to treat the disease. Because athlete's foot is caused by a fungi, antibiotics are not an effective treatment option here. We're now moving on to malaria. Malaria is caused by a protoctist called Plasmodium. It involves a vector. The word vector is used to describe anything used to transfer biological material, such as the bacterial plasmid used in genetic engineering to manufacture insulin, such as in the disease malaria. The mosquito is the vector because it transmits the protoctist, the Plasmodium, to the host, the human. Malaria is a dreadful disease. It causes millions of deaths. Even if it doesn't kill you, it causes such long-term illness that people are unable to work, which really affects the economy. Most of the avoidance of malaria comes from making sure the mosquito doesn't bite you, doesn't infect you with the Plasmodium. So how can you avoid that? Through use of mosquito repellent sprays, by wearing clothing to avoid the bites, sleeping under mosquito nets at night, again to avoid bites. You want to drain any stagnant water because this is a place where mosquitoes will reproduce. Insecticides may be used to kill the mosquito, and anti-malarial tablets may be used to kill the protist itself. There's no current vaccine for malaria, although anti-malarial drugs may be used to prevent the pathogen reproducing within the body. Another option is using sterile mosquitoes. These are mosquitoes which are unable to reproduce, therefore preventing the spread of malaria in this way, because there are fewer mosquitoes to bite people and pass on the Plasmodium. Typhoid, another waterborne disease, is caused by a bacterial cell. Again, it's spread in the feces of infected people, which enters the water supply. To avoid the spread, clean water should be drunk, hands should be washed after using the bathroom, and there's actually an effective vaccine which stops you getting infected with the typhoid bacterium. For people suffering from typhoid, oral rehydration therapy may be used as well as antibiotics due to it being caused by a bacterium. Houseflies have got a bad name for themselves in the spread of typhoid because they land on the feces of an infected person. They pick up that typhoid bacterium on their feet and then they land on food being left out on the side, transmitting the bacterium in this way. Therefore, food should be covered. So the bacterial cell, the virus, the protoctist has entered our bodies. How does our body act to fight against them? Well, first of all, the lymphocytes detect the antigens on the pathogen. They produce antibodies to help kill the pathogen. Some of the lymphocytes turn into memory cells. This means that upon re-entry of that pathogen, the memory cells can produce lymphocytes in a huge quantity, meaning that antibodies can be produced faster, sooner, and in greater quantity in order to destroy the pathogen before it takes hold. Antibodies are highly specific. That's because they have a variable region on them which binds directly to the antigen on the pathogen. The variable region is complementary in shape to the antigen. Through binding of the antibodies, it causes the pathogen to stick together, making it more easily recognizable by phagocytes, which then secrete digestive enzymes and engulf the pathogen. The antibodies also produce antitoxins against the toxins produced by the pathogen, and they can even cause the pathogen to burst open. If you want a more active approach to avoid getting ill in the first place, you might choose to have a vaccine. A vaccine is an injection containing a dead or weakened or attenuated form of the pathogen. What happens when that pathogen enters the body, it doesn't make you sick because it's dead or weakened, but your antibodies respond to the antigens on the pathogen. The lymphocytes produce memory cells so that upon re-entry of that pathogen, antibodies can be made again much sooner, much faster, and in much greater quantity. So vaccines are an excellent way of protecting yourself against a particular disease. Herd immunity is all about giving a large number of the population a particular vaccine. This reduces the number of potential hosts that can be infected by a pathogen, reducing the chance of this pathogen being transmitted. There's far less chance of a non-vaccinated person coming in contact with the particular pathogen when herd immunity has been used effectively. Talking specifically about vaccines, so I already said some vaccines contain a dead version of the pathogen, such as in the case of whooping cough. Some contain a weakened form of the pathogen, such as the case of TB and measles, whilst other vaccines simply contain the antigens of the pathogen, such as the case of the influenza vaccine. Now to discuss the difference between active immunity and passive immunity. Active immunity is when our body makes the antibodies itself, and that comes about due to infection by a pathogen or due to a vaccine. But either way, your lymphocytes will respond to the antigens present on the pathogen. They'll produce memory cells which, upon re-entry of that pathogen, make a huge number of antibodies super quickly. This is called active immunity because it's our bodies producing the antibodies. A second type of immunity is passive immunity. This is different. This is when antibodies are transferred into our body. It could be through breast milk when a baby breastfeeds. Antibodies pass from the mother to the baby. It could even be while the mother is pregnant. Remember, the placenta transfers antibodies from the mother's blood to the baby's blood, giving them protection from pathogens. But the key thing with passive immunity is that no antibodies are made by the person. And as a result, passive immunity gives you only short-term immunity as opposed to active immunity, which gives you long-term immunity. We now need to distinguish between artificial and natural immunity. So artificial, as the name suggests, is really vaccines. It's when you inject that weakened, dead form of the pathogen into your body, which causes the response, which I've mentioned many times now. Natural immunity occurs when a person has become naturally infected with a particular pathogen, when they've been exposed, for example, to the TB bacterium. But as with both, the pathogen will be recognized by the lymphocytes, memory cells will be made, large numbers of antibodies will be produced upon re-entry of the particular pathogen. Let's consider how someone suffering from Ebola may be treated. Let's take a person who has successfully recovered from Ebola. This means that their blood will naturally contain memory cells that recognize the Ebola virus. What happens then if you give a person who's suffering from Ebola blood from the person that's recovered? It means that the blood they receive contains memory cells which will respond to Ebola. Those memory cells will produce lymphocytes which make loads and loads of antibodies which will attack the Ebola virus in the person suffering, therefore killing that Ebola virus. This is a much quicker way of fighting the Ebola virus. It doesn't require the person's body to have to take the time to produce the appropriate antibodies, and this is currently a more effective treatment option than taking medicine. So as I've already said, antibiotics are highly effective against bacteria because they cause damage to the bacteria cell wall, which causes the cell to burst open through the movement of water by osmosis. But where do we get our antibiotics from? Well, two places really, fungi and other bacteria. Unfortunately, we're finding increasingly that the antibiotics we use to treat bacterial diseases are no longer effective. They are no longer killing the diseases of old. But why is that? It's because the bacteria are mutating. This is an example of natural selection, of course, meaning that some bacteria survive, they're no longer killed by the antibiotic, and because they survive, they reproduce, and before long, the whole bacterial population is now resistant to a particular antibiotic. The reasons why antibiotic resistance is on the increase is for the following reasons. People aren't completing their course. The antibiotics are being over-prescribed, particularly in agriculture. You find that antibiotics are given to animals even when they're not sick. And antibiotics are being given in cases when they're not even supposed to be used. Remember, I said that antibiotics are ineffective against viruses. One such antibiotic resistance case, which has been in the news a lot, is MRSA. Now, MRSA stands for methicillin-resistant Staphylococcus aureus. Staphylococcus aureus is the name of a bacteria which usually doesn't cause that much harm, that much concern, and that's because the antibiotic methicillin has historically always been very good at killing this particular bacteria. However, due to all the reasons I've just outlined, we're finding that Staphylococcus aureus is no longer being killed by the methicillin, and this bacteria, which previously was fairly harmless, is now actually causing deaths of people in hospital. And the reason for that is because people in hospital often have operations, they have open wounds, and this is a place where that bacteria can enter the body and cause much more damage. The MRSA is very difficult to treat now. Many antibiotics are ineffective against it, so it's becoming a real problem in hospitals. So how can we prevent antibiotic resistance? Well, it's the sort of things I've just been discussing, only giving antibiotics when absolutely essential, making sure that patients complete the course of antibiotics. They shouldn't cease to take the antibiotics as soon as they start feeling better, because there might be some bacteria still surviving, and these ones are more likely to mutate and become resistant, so patients must finish their course of antibiotics. We should be avoiding using antibiotics in agriculture, particularly when it comes to giving them to healthy animals. And it's obvious, but we need to have high hygiene levels in hospitals. As I've said repeatedly, antibiotics do not work against viruses. Obviously, we need to develop antivirals, but this is nowhere near as straightforward because the viruses infect our own cells, so it's hard to try and kill the virus without causing harm to the host cell. Decomposers are microorganisms such as bacteria and fungi which feed on dead materials, and decomposers have a huge role to play in treatment of sewage. Remember, sewage is human waste. The first step in sewage treatment is filtration. This is used to remove large particles. Sedimentation then occurs. This allows tiny particles to settle out. Aerobic bacteria and fungi oxidize any organic material. Anaerobic bacteria digest waste sludge. Methane gas is produced. Methane is an example of a biogas. Remaining leftovers can be used as fertilizer. And then chlorination is an essential process. This means adding chlorine to our water to make sure it's safe to drink, as it kills any pathogens present. So I've just described the treatment steps involved in a modern sewage treatment plant, but what about a pit latrine? As the name suggests, a pit latrine is a simple hole in the ground, which was used for hundreds, if not thousands of years previously. So a hole is dug in the ground over a sunken pit. Urine and feces drop into the hole and are broken down by microorganisms. Fly screens stop pesky flies reaching the feces and urine, and that therefore is important to reduce the transmission of disease, because remember I said if flies land on feces containing the cholera, typhoid bacteria, then they go and land on human food. This is a very easy way that typhoid and cholera may be transmitted. If sewage doesn't go after sewage treatment works and isn't properly handled, and say it washes into surrounding water sources like rivers and streams, what happens then is there's a huge increase in organic material in the rivers. Because the sewage contains a lot of nitrates, it actually causes algal bloom, so the algae in the water grow hugely. They block the light for the aquatic plants, meaning that the plants die due to a lack of oxygen. This provides food for bacteria, so the bacterial numbers increase, and because they aerobically respire, they use up all that oxygen in the water, meaning that it becomes anoxic, lacking in oxygen, so fish and other aquatic organisms can't survive. And remember as well that sewage contains an awful lot of bacteria in its own right, and this can actually increase the bacterial load so much in the rivers that kills the fish directly. Right, that brings us to the end of this all-in-one video. I hope you found it super helpful. It was a huge undertaking for me, so please comment below if you found it helpful. Do give it a like. Remember, if you've enjoyed the wording, you like how concise and exam-driven my answers are, you can find all this and more in my online revision guide available to purchase on the website swhlearning.co.uk.